Imagine a fragile plant exposed to the sheer violence of a winter snowstorm, surviving without a single pane of glass to protect it. The physical greenhouse has been replaced by an invisible barrier. In 2015, the Keshe Foundation introduced an agricultural framework designed to make physical shelters and chemical inputs obsolete. Their proposal substitutes traditional infrastructure with localized magnetic-gravitational, or Magrav, fields. The engine driving these localized fields is a proposed state of matter known as GANS, an acronym for gas in nano-solid state. Traditional agronomy relies on the continuous application of physical calories and chemical fertilizers. The GANS protocol abandons this cycle entirely, asserting that plants can thrive through passive energetic resonance alone. To understand how this is supposed to work, we have to look past the top-line claims and systematically deconstruct the hardware parameters, the precise loop geometries, and the biophysical mechanisms it demands. Regardless of how far this physics strays from the mainstream, the Foundation provides a highly specific, testable engineering blueprint, and that blueprint demands rigorous structural analysis. The entire Magrav agricultural system scales up from a single foundational building block, the closed loop. Constructing this loop requires strict adherence to dimensional and material constraints. Builders are instructed to use thin polypropylene tubing, specifically fixed at either 1/4 or 5/16 of an inch in diameter. There is a critical negative constraint here. Vinyl and other synthetic plastics are strictly prohibited, as they are theorized to cause severe field contamination. The injection sequence is highly specific. A narrow-peaked syringe is used to almost entirely fill the tube with distilled water, followed by a precise trace injection of the GANS material. Once filled, the tube ends cannot be tied off. They must be rigidly, mechanically coupled by sliding them into a slightly larger diameter sleeve made of the exact same polypropylene material. Heat fusing is explicitly banned, as altering the plastic's physical state compromises the field's theoretical integrity. The final product is not viewed as a simple plastic pipe. It is theorized to be a resonant containment vessel, one that relies entirely on strict material uniformity to function. Look at the intended output of the completed hardware. The loop acts as a generator, projecting a spherical magnetic-gravitational field. This localized field operates within a strict 3 to 10 centimeter boundary that perfectly envelops the local root system and immediate environment. The protocol offers distinct deployment flexibility, depending on the crop. The loop can be placed directly on the surface of the soil or buried entirely underground. According to the Foundation's spatial equivalence hypothesis, the orientation does not matter. The field's influence on the plant remains mathematically identical whether the tube rests in the open air or is packed beneath the dirt. Once the field is established, it can be calibrated for specific biological defense mechanisms. We can see this in application on Colombian coffee plantations, which frequently battle outbreaks of a destructive fungus known as Roya. To combat the Roya, growers deploy a specific variant known as Copper Oxide Gans. Copper oxide acts as a passive environmental disinfectant. It theorizes the eradication of the fungus solely through ambient field exposure, without a single drop of chemical touching the leaves of the coffee plant. The protocol does not treat the biological organism itself. Instead, it attempts to rewrite the spatial and environmental properties of the zone the organism happens to occupy. A single two-dimensional ring on the soil only protects a small radius. To shield larger crops, the protocol scales up the geometry into three-dimensional macrostructures. The vertical array scales this by stacking individual GANS rings vertically alongside the plant's stem, incremented exactly every 10 centimeters. This forms a cylindrical energy matrix, perfectly overlapping the fields to simulate the atmospheric containment of a physical greenhouse wall. For entire crop beds, the protocol transitions from single-plant vertical stacks to the frame array concept. These are large, overarching canopy structures that resemble standard clothes-drying frames. By piping GANS material continuously through the upper framework, a broad, overarching protective field is cast downward over the entire crop yield. The Foundation also claims these overhead frames absorb ambient moisture from the air, trapping the humidity within the localized field to create an autonomous, self-watering, and insect-free microclimate. By mathematically extending the loop geometry, the protocol attempts to render physical agricultural walls and roofs completely unnecessary. The effectiveness of these structures relies heavily on the specific atomic material utilized. Copper oxide Gans operates primarily as a biological disinfectant, but carbon dioxide, or CO2 Gans, performs a vastly different function. CO2 Gans is deployed specifically as a thermal regulator. It acts as an environmental shield against extreme temperature fluctuations. The Foundation cites Chinese agricultural experiments, where CO2 loops injected directly into freezing soil reportedly provided absolute frost resistance to the crops above. The most prominent thermal anomaly documented is this equatorial avocado plant. It survived an entire freezing German winter outdoors, completely undamaged. The hardware used for this specific plant was a thermos gradient. This is a complex layering of aluminum, copper, and CO2 Gans, specifically configured to accelerate the vortex of the field around the plant. The plant survived the entirety of the winter season without receiving any water and without any physical insulation covering its leaves. If these thermal repulsion claims hold up to scrutiny, it indicates that the CO2 material gradient actively overrides external climatic stressors, locking the organism inside a localized state of thermodynamic equilibrium. Validating such radical departures from standard agronomy requires incredibly strict experimental controls and relentless A/B testing. The Foundation's baseline requirement for any trial is the simultaneous monitoring of an isolated control plant, placed directly alongside a Gans-treated subject, exposed to the exact same environmental variables. Researchers utilize specific diagnostics like the protein shine test. By isolating a patch of topsoil beneath the field, they check for anomalous fat or protein extractions glistening on the dirt, generated purely by the surrounding ambient energy. Researchers also utilize condensation capture testing. Moisture dripping from the leaves of a treated plant is collected before it hits the ground to determine if the water itself has been transformed into a localized fertilizer. But the absolute extreme limit of the Foundation's claims pushes past plant life entirely into the concept of total biosustainment without consuming physical food. This limit was reportedly observed firsthand when a wild sparrow became trapped inside the Keshe Foundation's main industrial laboratory. The bird remained sealed inside the facility for 4 to 6 months. During that entire period, there were absolutely zero caloric food sources available within the building. The Foundation concluded that the sparrow survived because it instinctively drank from the open containers of Gans water stored on the lab tables, absorbing pure metabolic energy directly from the ambient fields. Concepts of pure energetic biosustainment clearly shatter the established rules of classical biophysics. Yet, the strict geometrically defined parameters of the Gans protocol provide a clear, falsifiable roadmap. It is an open invitation for anyone who is willing to test the absolute outer limits of alternative agronomy.